I'm now going to introduce Professor Cristina Sánchez, all the way from Spain. But this is an incredibly pertinent topic for a lot of people because it's mostly about breast cancer. This is medicinal cannabis and its therapeutic potential for cancer patients, in particular in the treatment of breast cancer. In 1971, Cristina graduated in biology at Madrid Complutense University. In 1994, she started her scientific career as an undergraduate student at the laboratory of Dr. Ramos and Dr. Fernandez Ruiz, the School of Medicine, Complutense University, where she first took contract with the field of cannabinoid research. Once graduated, she moved to Dr. Guzman's laboratory and the School of Biology at Complutense University, where she studied the effect of cannabinoids on lipid and carbohydrate intermediate metabolism first and on cancer cell proliferation later. Cristina obtained her PhD with honours in biochemistry and molecular biology at Complutense University in 2000. During her postdoc, Dr. Piamelli's laboratory, University of California, Irvine, 2000 to 2003, she studied the involvement of another group of bioactive lipids. These are the lysophil... I'm not even going to try and read that. It's complicated, you can ask her. And related compounds on pain initiation. In 2004, Cristina returned to Spain with Ramon Acaja contract aimed at repatriating Spanish researchers from abroad. And she started coordinating a new line of research with Dr. Guzman's laboratory. In particular, the goal of her research is to understand and exploit cannabinoids as a potential anti-tumoral agents in breast cancer. More recently, she has also focused her attention on new cannabinoid receptors and their possible involvement in cannabinoid anti-tumoral action in breast cancer and other types of tumors. It gives me great pleasure to introduce to you Professor Cristina Sánchez.
Well, thank you so much for the introduction. Apologize for the funny names. And I want to start by thanking the organizers for inviting me to participate in this event. Thank you very much. And I'm going to focus on cancer today. As you all know, cancer is a disease that is characterized by the uncontrolled proliferation of cancer cells, cells that have acquired mutations. And this process is accompanied by the generation of new blood vessels that irrigate these growing tumoral masses to feed them and to allow them to grow even bigger. At the final stages of this disease, new tumoral masses are formed at different sites of the original one in a process that we call metastasis, which is in fact the cause of most cancer-related deaths. So if this scenario is not complicated enough for a cancer patient, they also have to deal with other things like pain, in some cases associated to the progression of the disease, and with the side effects of the anti-cancer treatments they receive, which include minor things like hair loss, for example, but other serious stuff like anemia, pain, more pain, toxicity to major organs, and many other things. So we know from decades ago that cannabis can benefit oncologic patients. And they do it because they are very good analgesic compounds. And they are also very good in preventing some of these side effects of chemotherapy, nausea, vomiting, and lack of appetite. But what I'm going to talk about today is not cannabis as palliative care or in palliative care. I'm going to talk about the preclinical evidence we have showing that cannabis could be also used to fight against the tumors as anti-tumor therapeutic strategy. I'm going to go through this slide very quickly and just to show you that the endocannabinoid system we all have in our bodies, which consists of cannabinoid receptors, the endocannabinoid ligands, the ligands that we produce in our bodies that act on these receptors, and the proteins, the enzymes that produce these endocannabinoid ligands and degrade them. This endocannabinoid system that we all have control a wide variety of physiological functions, including, but not only, pain perception, motor coordination, the immune response, and many, many others. And going back to the oncologic context, this system controls what we call the cell fate. Cell fate understood as the very basic decisions that cells take during their life, which are basically whether to proliferate and generate more cells, or whether to differentiate and become committed cells, become astrocytes or neurons or hepatocytes, for example, or whether to die. And this is a very physiological, normal decision. Cells can decide to die. And after many years of research, we know that what cannabinoids do, what the activation of the endocannabinoid system does in cancer cells, and only in cancer cells, is activating this cellular program, cancer cell death. So when one is dealing with a potential anti-tumor compound, the first thing that it has to prove is that it has anti-tumor properties. And you have to do that at different levels. We always start with very basic experiments in cell cultures, and if we get positive results there, then we move to something a little bit more complex, which are animal models of cancer. I will give you more detail on the different options we have there. And if we get positive results there, then we pass the torch to our colleagues, the clinicians, and they do studies on humans. In the case of cannabinoids, we followed this exact same path, and we started 20 years ago by doing basic cell culture experiments. We challenged cancer cells cultivated in plastic plates with different cannabinoids, at that time THC. And what you have in this slide is what we got at that time. On the left-hand side, you see glioblastoma cells. It's a type of brain tumor, extremely aggressive brain tumor. And these cells are growing happy and healthy in the plastic plates. And what you have on the right-hand side is these cells after THC treatment, 24 hours. And you see fewer cells, and the few that are left are detached from the plastic plates, round and refringent, and these are all signs of cell death. We observed that in glioblastoma cells many, many years ago, but from that time, different groups, not only ours, have been working on cancer cells and cannabinoids, and this is the picture you will get if you treat cancer cells with cannabinoids. You will need a little bit more of THC, a little bit less in some cases, more CBD, less CBD, but you will end up having a picture like that. So we next decided to analyze whether we were able to reproduce this effect in an in vivo setting, in an animal model of cancer. And we started by using the model you see on the left. This is what we call ectopic xenograft. We like funny names, difficult to pronounce to make your work complicated. So we call it ectopic xenografts. What we do here is to inject cancer cells underneath the skin of immune deficient mice. So the animals generate tumoral masses. In this case, it's a brain tumor in the side of the mouse. So we treat these animals, and this is an example of what we get when we use these kind of models, not only in GBM, not only in glioblastomas, but in many other types of cancers. We observe a reduction in tumor growth, and you can see that comparing the size of the tumoral masses in this animal here that did not receive THC treatment with the size of this mass here, a mouse that received THC treatment. Or comparing these images here, where you can see that the tumors are not only different in size, but also in color. You can see that the tumors that have received THC treatment are paler. And that means that these tumors are not irrigated. So we are blocking not only tumor growth, but also angiogenesis, which, if you remember, is the process that allows the tumors to grow. One of them, not the main one, but one of them. But when you think about the patients, they don't have brain tumors in the side. So the next thing you can do is to generate the tumors in the exact same locations where the cells were originated, where the cancer cells were originated. So in this case, in the right part of the image, you see brain tumors generated in the brain of the rats in this case. So we were able to inject brain cancer cells in the brain of these animals. We have done the same thing with breast cancer cells, injecting them in the breasts of the animals. And we have observed the very same thing that we did with the ectopic models. This rat here has a brain tumor. This white mass in the MRI scan is the tumor we generated. And on the right, you have the very same rat after cannabinoid treatment, after THC treatment. And that mass has completely disappeared. But again, when you think about the human pathology, we don't have tumors because someone injects us cancer cells. Our tumors appear spontaneously. And we were lucky enough to have access to models of cancers that appear spontaneously. In this case, we moved to breast cancer, and specifically to a subtype of breast cancer that is characterized by the overexpression, by expressing huge amounts of a protein that is called HER2 here. HER2 is an oncogene, a protein that makes cells proliferate without control. And these animals have the peculiarity that have been manipulated genetically to overexpress the rat version of this protein, which is called NEU here. So the consequence of this manipulation is that the females, and only the females, when they reach adulthood, they develop breast tumors, and they do it spontaneously. We don't have to inject any cancer cells, they do it spontaneously. And they do it after very long latency periods. In some cases, it takes half of the life of the animal to generate the tumor. And in about 70–80% of the cases, the animals end up developing lung metastasis. For all these reasons, this model is considered not only by the preclinical community, but also by the clinical community, a very solid model of HER2-positive metastatic breast cancer. So what we did with these animals was to treat them with THC for 3 months. And we observed, first, that the growth of the tumors was reduced. You have in this graph here on the left how the tumors grow in the group that has not received THC treatment. We represent tumor size, tumor volume versus time, and you see that the tumors grow exponentially. But when the animals receive THC treatment, this growth is significantly reduced. But not only that, THC also reduced the process of tumor generation. I don't know if you know this, but mice have 10 breasts, and each one of them is susceptible to generate a tumor. And what we observed was that in the control group, in the group that did not receive cannabinoid treatment, 41% of the animals developed 4 or more tumors. But in the vehicle-treated group, none of the animals presented 4 or more tumors, and half of them, 50% of them, presented just 1. So THC was reducing not only tumor growth, but also tumor generation. And something that is very, very important, because as I said before, the cause of death in most cases, is metastasis, not the primary tumor. THC was also able to reduce the percentage of animals that developed lung metastasis. This number was roughly 70% in the vehicle-treated group, and went down to less than 20% when the animals received THC. And after many, many years of work, we know, and this is very important for us, what is going on inside the cells, inside the cancer cells when they are exposed to cannabinoids. We know the molecular mechanism by which cannabinoids are killing cancer cells. We know that cannabinoids, and especially THC, bind and activate CB1 or CB2 receptors, and it depends on the type of tumor. It seems to depend on the type of tumor, if CB1 or CB2 is the main actor in the movie. This activation produces a cellular response that we call the endoplasmic reticulum response, which modulates the expression, the amount, and the activity of a number of proteins inside the cells. You have the numbers here, but I don't want you to waste any single second to try to read them. I just want you to focus on this protein here, AKT. All this information, all these changes in proteins inside the cells convert in the inactivation of this protein, AKT, which is the master regulator of cell survival. So cannabinoids switch off the protein that controls cell survival. And this inactivation triggers two very interesting processes inside the cells that are called autophagy, which means self-digestion, and apoptosis, which is the fancy way to say suicide. So in summary, we know that cannabinoids inactivate AKT and induce cancer cell death by apoptosis. And this happens not only in glioblastoma cells, but also in breast cancer cells and in many other different types of cancer cells. At least the three main features of this molecular mechanism that I just described occur in basically all the cancer cells or cancer models that have been studied. So we know that THC blocks cancer progression, acting at different levels in our preclinical models of cancer, but THC is not the only cannabinoid with anti-tumor properties within the plant. Another very interesting compound that produces the same effects is CBD, cannabidiol. And we know a little bit less about the mechanism of action of cannabidiol in cancer cells, but we also know that it produces apoptosis, cancer cell death, by activating reactive oxygen species or inhibiting AKT as well. We know that it also blocks angiogenesis, and we know that it also blocks metastasis. And the interesting thing is that these effects are produced by different mechanisms of action. And this is very good because when you combine THC and CBD, you are attacking the tumors from different sides. And the response, the final response, is greater than the one produced by each of the compounds separately. This is just an example, but it has been observed in many different types of cancer. You have in this line, the white one, how the tumors grow when the animals do not receive cannabinoid treatment. In green, you have the response to THC, a decrease in tumor growth, very similar to the one that produces CBD in yellow. And when you combine THC and CBD, the effect is greater. And cannabinoids combine well not only with themselves, but also, and this is very important when we think about the patients, with standard treatments. They combine very well with chemotherapy. And this is just an example of the effect that we obtain when we combine cannabinoids, in this case THC, with temozolomide, which is the standard medicine for GBM, for glioblastoma patients. When we combine THC, that produces a mild effect here because we were using very low doses, and temozolomide, we practically get rid of the tumors completely. And cannabinoids also seem to combine well with radiotherapy. This was published in 2014, and in this case they combine cannabinoids that produce no effect or very low effect because the doses were very low with radiotherapy, and as you can see, the combination dramatically reduced tumor growth. So we know, at least at the preclinical level, that cannabinoids work as anti-tumor agents. But we also have to prove that they are safe. And we have to do it at exactly the same levels as where we proved efficacy. So we started by working in cell cultures, and we observed that cannabinoids have nothing to do with chemotherapy. And I mean that cannabinoids seems to act only on cancer cells, not on cells undergoing proliferation, as chemotherapy does, but only and exclusively on cancer cells. And this is just an example. I'm showing you just examples of the things I'm mentioning. In this experiment, we treated different cancer cells, breast cancer cells, with THC, and the cancer cells are represented in blue and in green. Here I'm representing how much the cells die, or the percentage of the population of cancer cells that die in response of THC. So, no, sorry, I'm representing the percentage of cells that remain alive when I treat the cells, I'm sorry. So as you can see, when we treat the cells with THC, the breast cancer cells in green and blue, they all decrease their proliferation in response to this cannabinoid. But in yellow, we have non-cancer cells, breast healthy cells. And these cells do not respond the same way to THC. They don't receive the signal to die. They don't die in response to THC. And this is the case not only for breast cancer, but for any other type of cancer cell line that has been studied. And we have in vitro proof that cannabinoids are safe, and we also have in vivo proof that they don't produce any toxic effect. Every time we do an in vivo experiment, an experiment with animals, we send the organs to our pathology, and she has never ever detected any sign of toxicity at all. But in this case, we also have clinical information saying that cannabinoids are safe, that cannabinoids are safe medicines. If we go to this site, clinicaltrials.gov, which is an NIH site, and we go and find search for the clinical trials that have been completed with cannabis medicines, we will find about 500. And they are all pointing in the same directions. These medicines are safe. Well, the answer we have, the question we need to answer is not if cannabinoids can cure a mouse that I have in my lab. The question we need to answer is if cannabinoids work as anti-tumor agents in humans, in actual patients. The only information we have so far about this is a small clinical trial, a small pilot clinical trial, very small, just nine patients, that was performed in a hospital in Spain in 2006. 9 GBM patients were enrolled in this trial, and we observed that THC produced no physical, neurological, biochemical, or hematological effect on these patients, and that THC worked as efficiently as temozolomide in reducing tumor growth and increasing the length of survival in some of the patients. I want to stress out that we were working with 9 patients, just 9 patients, so the conclusions are not very solid, but this is what we observed. And something that is very important for us is that THC seemed to modulate the same molecular mechanisms that we described in our preclinical models. THC decreased cell proliferation. You can see here that in the right image there are less yellow dots. The yellow cells are the ones undergoing proliferation, and you see that cannabinoid treatment decreased the number of cells undergoing proliferation. THC also reduced or blocked the angiogenesis process. There were less blood vessels in the tumors of the patients that received THC. And THC was also inducing apoptosis, as you can see in this slide, comparing the number of brown dots in the 2 images. And this is very important for us because, again, this is validating the preclinical models we are using in the lab. This trial was performed in 2006, and after that, nothing happened. Until very recently, when GW Pharmaceuticals started a clinical trial in the UK, aimed to analyze the efficacy of Sativex in combination with temozolomide. The recruitment period has already finished, and results are expected for the end of the year. I found another trial in this website I mentioned before that was going to be performed in Israel, in this case with CBD, in patients with solid tumors, but unfortunately the trial has not started yet. I don't know why. but hopefully they will start soon. But these are the only clinical trials that are in progress right now, which is absolutely, I don't understand why. So just to sum up, I think we have more than enough preclinical evidence showing that cannabinoids produce anti-tumoral responses, at least in our preclinical models of cancer. We have a lot of information saying that cannabinoids are safe medicines for patients. We know that they combine very well with standard anti-cancer treatments. And we also know, and please don't forget this, that cannabis has additional advantages for cancer patients. They are analgesic, they stimulate appetite, they prevent nausea and vomiting. So what do we do next? Well, we in the labs will keep on working on our preclinical models. We want to know more about the mechanism of action of these compounds. We want to know more about how they combine with therapies, because the better we know how they work, the better we can target the tumors. But what we need at this point is more clinical information. And we need clinical information that can come from many different sources. We can do clinical trials, randomized clinical trials, but we can get information from other sources. We have places in the world where medicinal cannabis is legal or is about to be legal, and these patients will be and are already using cannabis. So let's collect that information and show it to the scientific community, to the medical community, in a good way. Thank you. And I just want to finish by thanking the people that have been doing most of the work. I'm just presenting it, but the work is actually being done by other people. People in our lab, and I want to mention especially the people in my lab that is working on breast cancer. Sandra is sitting over there. She's doing a short stay here in Australia. She's doing research here in Australia, and she's sitting right there, so you can ask her everything about our experiments. And thank you very much for your attention.